thank the organizers very much for inviting me. It's a very interesting conference. So I'm going to talk about a very new venture we've taken on in the lab. My lab studies the retina, and I'm interested in visual processing of the retina. But very recently, we've been trying out a new technology, directing a novel sensory input, ultrasound, to the retina. And so this work that I'm going to talk about is in collaboration with ultrasound engineer Pierre Courier-Coub. And to tell you a little bit about the recently developing field of ultrasound stimulation, it's been known for quite some time that you can change activity in the retina using ultrasound. This is actually known since 1929. This is a paper from 1958 showing that if you direct ultrasound to the LGN, you can change neural activity. In this case, they're suppressing neural activity. It's a very slow modulation. Here's a visual response. It goes down in the presence of ultrasound, and then it recovers afterwards. So people didn't really pay attention to this very much for some time. In 2008, Jamie Tyler, who's the next speaker in the session, shows some very interesting work showing that if you direct low-intensity focused ultrasound to, this is a brain slice preparation, you can actually generate very precisely timed neural activity. So the arrow shows the time of the ultrasound, and spikes are generated, and they characterize this effect. So I think this set off a great deal of interest in being able to use ultrasound to change neural activity to precisely deliver neural information to the brain, in the same way that other techniques, artificial techniques, have been used, such as electrical stimulation, magnetic stimulation, recently optical stimulation. So we came into this a few years ago, basically by urging of Bill Newsom at Stanford, who was aware of this work, and actually he's now on the Brain Project Committee, President Obama's Brain Project Committee, sort of to decide what are should be the important goals of neurotechnology and study of the nervous system. And so he urged us to consider this. And so we started talking with some of the ultrasound engineers at Stanford, in particular Pierre, and thought about what would be necessary to try delivering ultrasound into the retina. So what we do, as I mentioned, we study the retina. So here's a diagram of the vertebrate retina here. So we take the retina out of an animal intact, typically a salamander or a mouse, and I'll be talking about salamanders today, even though all vertebrate retinas are basically the same cell types and the same types of responses, and we think any result in one retina will really likely transfer to another. So the sort of experiments that we usually do is we take the retina out of the animal, and we place it on a flat electrode array, so we can record some dozens of retinal ganglion cells, cells that deliver information to the optic nerve. And so we do things like deliver visual stimuli either from above or from below. We also do intracellular recording and optical imaging. As I say, our usual questions are we want to understand how the retina is processing visual information, how the cells and synapses are performing neural computations. So it turned out that Pierre's lab had a transducer that looked basically just like a microscope objective and fit right into our setup. And so here's an example of the data that we would record, but let me just talk about the setup first. And so one of the nicest thing about that is that we didn't even have to change the schematic. We just changed the label on the schematic, and it fit right into our setup. So the sort of data we get out of this recording is shown here. So each line is a different ganglion cell here. This is a 4-second response this is a visual stimulus. And so what we did is to use focused ultrasound, and this is the type of stimulation that we use. This is a 43 megahertz transducer. It's focused. The size of the focal spot, given the frequency, is about 90 microns. We wanted a spot that was smaller than the receptive field size of a ganglion cell, so we could measure its effects. We're stimulating throughout the entire retina on this scale. The retina would look like this. It's much wider than that, but we're stimulating throughout the entire depth of the retina. We're stimulating all layers. The modulation of the ultrasound carrier frequency that we use is low frequency, essentially in the range of visual, typical visual stimuli. So when we compare the visual stimuli to ultrasound stimuli, so here's what we saw. So here's the stimulus where we flash a light on the retina. This is a spot just about the size of the ultrasound stimulus. And we saw very reproducible spike trains. This is what we usually see. This is a typical response of a retinal ganglion cell. It's firing at both the onset and the offset of light. When you turn the ultrasound transducer on, we saw these responses. The responses were very precise, very reproducible. The recordings would last for hours. Basically, the recordings lasted longer than the experimenter did, and the experimenter gave up. And for all intents and purposes, if you showed these recordings to visual neuroscientists, they wouldn't think these are remarkable. As you can see, there's some differences that we saw. The responses to ultrasound were of shorter latency than the visual stimuli. Here, the latency compared with visual stimuli and ultrasound stimuli. And remember, these are in the exact same neurons. You can give the visual stimulus and then the ultrasound stimulus afterwards. And in fact, the jitter, the variability was just the same or even a bit less than visual stimuli. Actually, this told us something interesting about retinal processing right away, because we think at least the ultrasound will be skipping phototransduction. It's thought that a lot of noise in the retina comes from phototransduction, and because we're skipping that stage and the noise doesn't really change very much, under these stimulus conditions we can conclude that most of the noise is actually coming downstream of the photoreceptors. So we were sort of very surprised by this and encouraged by this, and it was clear. This is the first time we tried this. This worked. Worked very clearly. And so we considered that this would be something, essentially a real technique that we would use. So what would we use it for? So the things that we were thinking about were really twofold, maybe threefold. So one was as a basic science tool to be able to characterize the cells and the connections. If we could arbitrarily access individual cells, we could learn something about their processing. A second thing we wanted to learn was something general about this artificial stimulation technique. The retina is a very controlled prep. We understand a lot about it. We can give its natural visual stimulus. And so, as a way to characterize the effects of ultrasound, we understood that this was going to be useful for anyone that was going to be using ultrasound in any part of the brain. For example, going through the skull, where it might be a little bit more difficult to understand exactly what the processing is going on in circuits of the higher brain. And the third thing we're thinking about is as a potential for a retinal prosthesis. If in cases of photoreceptor degeneration, as you heard earlier today, in the case of electronic retinal prosthesis, potentially ultrasound could be delivered through the eye, and it would be a non-invasive retinal prosthesis. So we then wanted to characterize the effects of ultrasound more thoroughly. So this is just a simple flashing light. The way we usually characterize the responses to visual stimuli is by making a quantitative model of the response to tell us exactly how neurons are encoding the stimulus. Here's an example of a typical experiment in a model. So we use a randomly flickering light, here flickering on and off, and then we calculate a model that's a simple linear filter, an impulse response function followed by a threshold nonlinearity. What this linear filter means is that on average, a photon is producing this change in firing rate, or symmetrically, you can think about this effect as this stimulus the cell is most sensitive to is when the light turns on and then turns off. The nonlinearity is a function that characterizes properties such as threshold and sensitivity and any saturation. So we did the same thing for ultrasound. So we modulated the ultrasound stimulus in the exact same way, at the same high temporal frequency at 30 hertz. And what we saw was that we could compute filters for ultrasound stimuli and thresholds just as we could for visual stimulation. So this tells us clearly that we can deliver sensory information using ultrasound at a high bandwidth. You see that the temporal filter is a bit different than the visual filter. And that was an indication to us that we're probably skipping more than just phototransduction. We'll tell you a little bit more about that later. The effects varied in different cells. Here's a cell where the responses just basically look speed up in latency, but are the same time course. Here's one where the time course is different. Overall, we saw that the peak frequency of the ultrasound filter was higher than the peak frequency of the visual stimulation. So sort of going on with this shorter latency, it looks like we can actually convey faster temporal information using ultrasound than visual information. We then ask a different question, which is one that one is not often asked in the case of artificial stimulation. You might be able to measure that an artificial stimulus might activate a cell, but if you're delivering that stimulus to some part of the brain that still is receiving input, you really want to know what it's doing and how it's responding to its own natural ongoing input. And the reason that's important is because if you don't know what that effect is, even though you may activate a cell, you may actually be dropping its sensitivity to its own natural input or raising its sensitivity to its own natural input. And you could actually make sort of sign errors about what that part of the brain is actually doing. So the way we measured this effect is by modulating a visual stimulus over time and then pulsing ultrasound every second or so. And then again, computing properties of the neural code at different times when we do this. So in response to the pulse ultrasound, we saw firing rate changes here, smaller and then greater, and returning back to a baseline. If you compute linear filters at these different intervals of time, these don't really change very much. So ultrasound didn't affect the ongoing temporal processing of the natural visual stimulus. The sensitivity to ongoing visual input changes. It either lowers here or raises here. But one thing that we see, it very rapidly returns to baseline. So there's not really any prolonged shutting down of the retina in response to brief ultrasound stimulus. It recovers very quickly. These effects are somewhat varied across cells, and we think that's because the different types of cells have different types of neural circuits that feed into them. So there was different changes in gain across different cells and different changes in threshold, but we thought that's generally to be expected. So we thought that the effects are localized in time. Then we asked whether they're localized in space. To do this, we looked at the combined receptive field maps of many ganglion cells, and by pooling those all together, we can sort of measure an overall spatial map of what the sensitivity to the visual stimulus is. And so these are just the cells that we happen to record from in the experiment. Then we could make a difference to this map of sensitivity when we're stimulating with ultrasound or not. And so when we made that difference map, what we saw was a very narrow focal spot where the sensitivity changed. And that narrow focal spot was about the size of the ultrasound focus, about 100 microns or so. So what this told us is that if we're stimulating one part of the retina, we're not causing very wide field effects, damaging cells that have processes feeding through this area. The effects are very localized in space. And so we think that's important to the study of neural circuits, and also important for a potential application as a prosthesis. We then wanted to consider what the possible mechanisms are for ultrasound. So the first type of mechanistic effect we considered is what is the physical effect? Types of physical effects that have been proposed are what's called radiation force or radiation pressure. This is the nonlinear effect of radiation being attenuated, absorbed, or reflected at a change in medium, very much similar to photon radiation pressure. If photons are absorbed or reflected, they create a force. Ultrasound, if it's also absorbed, it will also create a force. Cavitation has been proposed, either extracellular cavitation. We don't really think that's an issue because of the high frequencies that we're using. An interesting proposal has been what's called an intramembrane cavitation proposed by Eitan Kimmel. And essentially the idea is the drop in pressure caused by each cycle of the ultrasound causes the membranes to briefly separate, change in distance, and that causes a great change in capacitance. And this is only proposed on theoretical grounds. We don't really have evidence, or anyone has evidence of this yet. Temperature change we think is minimal because we've measured changes in temperature and they're well sub-degree. So to consider what we did was an experiment to image what's going on in the retina when we stimulate. So this is a fluorescence image. The dye is FM464. We're measuring with a 2-photon scanning microscope. We're scanning while we're stimulating with ultrasound, and the scanning is uncorrelated with the ultrasound, and then by doing many scans and averaging them, we can create essentially a millisecond time scale measurement of what happens on average in response to an ultrasound spot when it's flashed on. Okay, so here's the movie. So here's what we saw, and basically this is what we saw the first time we turned it on. So you can sort of first see that there's some intensity changes that this stimulus shows when the ultrasound turns on. The spot is this size. There's intensity changes that you can see in the center here. That's because the retina is basically being pushed down from one focal plane to another. And so out here you can clearly see it looks like kind of the retina is breathing. There are lateral shifts that are going on here on the scale of a few microns. So what this suggests to us, and Pierre has modeled this, that there's definitely a measurable radiation force that's occurring. It's consistent with, these effects are consistent with what you'd expect from the radiation force, both the displacement in the axial direction and the shear as well are consistent with radiation force. It's not to say that this is necessarily the explanation, but it's consistent with those effects. If you take a slice through this image here and show it in XT, you see here, this is the ultrasound going on and off. These shifts here are the lateral shifts that I just showed you. They go in opposite direction at the focus. Here it moves up, here it moves down. If you measure the displacement immediately after the ultrasound turns on, we see a displacement that's as fast as we can measure, less than 5 milliseconds at a 25 millisecond bin time here. And then there's a slow decay of about 100 milliseconds that we think is just the tissue essentially relaxing. Okay. When we compare in different preparations the size of the displacements that we observed, and compare that to the amount of neural activity that we observed, we see that there's a very similar curve as a function of power here. So it again shows these two things are highly correlated, and we think that this displacement is maybe from radiation forces is perhaps the underlying mechanism that generates the neural activity. Okay, so. As to what are the biophysical effects, okay, so it might be radiation force, but what is it doing? Is it changing the capacitance? Is it causing effects on mechanosensitive ion channels? Another interesting hypothesis is that it may be affecting synapses. So people haven't measured synaptic vesicle fusion as a function of ultrasound pressure, but there's an interesting paper from Erwin Nair's lab showing that if you put a whole patch clamp apparatus in a pressure bomb and you pressurize it, synaptic vesicle fusion is highly sensitive to pressure. So what we did was to pharmacologically block synaptic transmission. And so in response to a flashing light or ultrasound, we see activity in this cell, and then we blocked all synaptic transmission with cadmium chloride, and basically we saw activity go away. And so what that tells us is that we're not primarily stimulating ganglion cells directly, because if we did, once we blocked all synaptic transmission, we'd still see the effect. So the effect seems to be requiring synapses. So it suggests to us that so even if we turn the power way up, right, we don't get direct activation of ganglion cells. So maybe synapses are required. So potentially we are considering direct effect on synapses as a mechanism. So I want to talk now a little bit about the potential of ultrasound energy delivery for a retinal prosthesis. As we heard earlier today, there's a number of diseases where the photoreceptors degenerate, but the rest of the retina is still there. It's not the same, it rewires a bit, but it's still there. Retinitis pigmentosa is in about 1.5 million people worldwide. Age-related macular degeneration, 30 million people worldwide, particularly in older Americans. So we heard something about the state of the art in electronic retinal prostheses earlier today. But one thing that's common about them is that they're invasive. You have to put something in the eye. And in particular for older patients, considering undergoing a surgery may be a big issue. Another thing to consider is that if you're going to put a retinal implant somewhere, even an electronic retinal implant, if the photoreceptors are gone, there's no way to diagnose whether there's any surviving retinal tissue there or what's the best place to put the retinal tissue. And so we've actually thought a bit about retinal prosthesis. We've collaborated a bit with Daniel Palanker's group at Stanford, who's worked on a high-resolution photodiode retinal implant. So we've thought about things like image processing. So no matter whether ultrasound is a substitute for an electronic retinal implant or just an initial test bed for future electronic retinal implants, it's something that we're thinking about. So a critical aspect of this is that we want to make sure that the ultrasound stimuli are stimulating cells beyond the photoreceptors, because if they're only stimulating photoreceptors, it's going to be less useful for a retinal implant. So what we did was to take advantage of the fact that the bipolar cells in the retina come in two types. They're on bipolar cells, they respond when the light go on. Off bipolar cells, they respond when the light goes off. There's a very specific and well-characterized drug called LAP4 or APB that blocks the metabotropic glutamate receptors, blocks the specific glutamate receptor that feeds into the on bipolar cells. And these effects are well understood. So since we know that many many cells respond to ultrasound stimulation at both the onset and the offset of the stimulus as light does, we then blocked just the on pathway and we observed what happens to the light and the ultrasound stimulation. So we observed, first of all, that as we expected, light blocked visual stimuli going on. See these spikes here, this big black peak that's hiding behind the red one disappears here. And so, as expected, LAP4 blocked the on pathway, but it didn't do so in ultrasound stimulation. So if ultrasound was only stimulating photoreceptors, then the drug that blocks the on pathway would have the same effects in ultrasound stimulation as visual stimulation, because it blocks the receptor side, the postsynaptic side of the bipolar cells. Across a population of ganglion cells, if you compute an index that tells you how much the on pathway was blocked, it wasn't in ultrasound. So this tells us that to some extent we're activating cells beyond the photoreceptors. Okay, so what would a retinal prosthesis look like? So we've thought about this, and mostly Pierre has thought about detailed designs. And the design that he's conceived is essentially a contact lens, where there's an array of transducers, and Pierre, who's often thinks about CMUTs as a capacitive micro machine ultrasound transducer as a transducer of choice. I think he's thinking about one of these as a possible transducer, or an array of these that sit on a contact lens and can deliver focused acoustic information onto the back of the eye. So in the case of a disease where there's no visual sensitivity anymore, the array could cover the entire contact lens. But if there is visual sensitivity, as in the case of macular degeneration, then an annulus of transducers would preserve any intact visual information. This is a simulation that Pierre just gave me about what an array of 64 by 64 2D a 2D array at 25 megahertz would look like, and just to demonstrate that it could arbitrarily create a pattern, multiple foci in two spots next to each other at a distance of about 25 millimeters, which would be about the back of the human eye. So in terms of transducer technology, this appears to be doable. In terms of the biology and what are the effects in patients, of course, that remains to be seen. So, in summary, we've learned a lot about focused ultrasound use in the retina. It can be used to continuously deliver information at high bandwidth. We've learned something about the mechanism. It creates micron-scale displacements, and synaptic transmission is required, which is implying to us that maybe it is a direct effect on synapses, although certainly there may be multiple effects, and also multiple effects in different systems. So we've learned that for basic studies, we can measure the effect of pharmacologically defined neural pathways, and it creates localized changes of sensitivity to its natural input, which we need to be aware about when interpreting the effects of ultrasound. As far as we know, single-cell stimulation is feasible. So 43 megahertz translates into 90 microns or so. If we go up to 10 times higher the frequency, we may be able to address single cells in a kind of random access way. And finally, we think it holds promise for a translational use. It activates cells beyond photoreceptors, and we think it has potential for a non-invasive retinal prosthesis. So to acknowledge the people who did the work, Mike Mentz in my lab did all of the retinal experiments, Pierre's postdocs, Omer Al-Ralkan and Amin Nikuzadeh did the ultrasound engineering needed, and we've also collaborated to mention with other faculty at Stanford, Kim Butts Pauley, thinking about in vivo stimulation, and also Merrick Madouk thinking about the biophysics of retinal stimulation. And in terms of funding, I'd like to mention Stanford NeuroVentures, which is essentially sort of a Stanford foundation in some way. It gathers private investments together and delivers them to very novel projects like this one. And I want to thank you.
Very interesting, thanks.
It's really interesting. On the cadmium experiment, is it possible that a cell upstream of the ganglion cell is being preferentially activated, and that therefore that's what's so I think your idea was that you were stimulating the synaptic vesicles, but if instead you were stimulating some other population of cells preferentially to the ganglion cells, that would also account for. Is that still in the realm of consideration?
That's absolutely possible, right? It doesn't. It seems to be somehow a cell non-specific effect. That's formally what we can conclude. And so all cells have mechanosensitive channels, and so it's not a set of channels that are in all cells. But for example, with bipolar cells have a different set of channels. There's nothing to do with the synapse.
But I guess where I was going with that is that one sort of intriguing and exciting possibility would be that you could preferentially stimulate different subsets of neurons by tweaking the ultrasound characteristics. And I'm wondering whether there's a possibility that you actually already have a signature of that.
Possibly, right. So let's say it's not just bipolar cells that are more sensitive. There's a think of the sensitivity profile, preferred feature for ultrasound stimuli, basically. And I would say it's not only in terms of channels. You can think of it in terms of timing, in terms of those temporal filters. Can you design a stimulus? Yeah, I think that's potentially possible. So we have plans to do single cell. We want to do calcium imaging, so we want to measure direct effects, and then we can characterize that better and decide whether or not we can tweak it.
So are you thinking about mechanical thing, like there being mechanical resonances that you want to get? Amplitude modulation of the...
Potentially, yeah. So if there's some set of mechanosensitive channels, then everyone will have some preferred stimulus, whether it's a temporal frequency or.
So in the early 1950s observation that ultrasound depresses neuronal activity, does that fit with any of the models about what?
Yeah, I'm not sure about any of those old results. I don't know if Jamie, you want to comment about that. Do they use like 100-second stimuli, and I don't remember what the power levels?
I don't think they said in that particular paper. It's high. It's assumed to be thermal, right? I think the early stuff was all assumed to be thermal.
Yeah,
Is there a sort of a two-photon equivalent of ultrasound, nonlinear effect that you could use for maybe sectioning or improve your points per function?
yeah, two-photon.
something non-linear.
There's some diffraction limited ultrasound for sure. You can use metamaterials, acoustic metamaterials to get. You can definitely beat the diffraction in most ultrasound. So two-photon microscopy doesn't necessarily beat the diffractions, but it will allow you to achieve better imaging than the standard.
Why do you use that? And then you have better spatial resolution.
That technology is very new.
Yeah, I don't know if Pierre has a comment on that. It's an interesting idea. It's something we'll probably engineer about.
I believe there are people here who know something about that.
Yeah.
Maybe we'll hear some about that tomorrow. I don't know.
I was curious about the filters you calculated. You said if you calculate the filters responding with respect to light input versus sound input, differences. And if you calculate the same filters with respect to direct current injection, do they then map more closely to the filters judgment of sound?
Interesting. So the question in general is what builds a filter, and the linear filter is a combination of essentially everything that went from the stimulus to the response. So in the case of if you injected current, would you actually find that signature? Can you count all the properties of the filter by current injection that would imply that it's direct stimulation? It's a good experiment. We haven't done that yet. One thing we basically could do that. So what we do is if you want to measure a filter, maybe you want to do it with an optical image, so you can really measure cell that the filter of the cell you're recording from. We don't think we're still in ganglion cells, right? So you want to measure the filter of the cell that you're stimulating. So let's say we measure the filter of ultrasound of a bipolar cell. And instead you'll enter in that bipolar cell and inject current. And we
Tim?
I was intrigued with your temporal response from the radiation force experiment. If I understood correctly, you had about a 5 millisecond sampling interval? What was limiting
it was
that?
25 millisecond, and so that, really to reconstruct that series, the way we're doing is we're scanning for many trials, and then whenever the ultrasound pulse is on, we keep a track of where we was, so where we were. So we're not imaging at 25 milliseconds, but we can reconstruct at arbitrarily fine time resolution. So it's basically limited by a single noise ratio. You probably get down to 5 milliseconds. So what I showed you there was an analysis from 25 milliseconds. So it's essentially a change in one frame, and so that's what we're saying it's time constant less than 5 milliseconds. But we could conceivably, especially if we're scanning not a frame but a smaller area, go down to an arbitrarily fine time resolution. So yeah, we have a comment about that very.
I was just wondering if there was information that would be useful to you in that short temporal response.
Oh yeah.
And it sounds like you can sample different ones.
Absolutely, yeah.
Because when you pushed on it, that should have induced a transverse wave that you could attract that may have excited things next to it.
Yeah. Yeah, so absolutely that would be of interest, and so we just need to do the experiment to pull that out and then peer with model the effect and see if we can understand.
And at least visually, it looked like whatever damping, mechanical damping in the tissue was very fast also. Right? You didn't see a lot of, almost prompt response and relatively little ringing. There was
Right, exactly.
And so I would caution though that even with good signal to noise, if essentially using my language, you want to split a line, you want to take something where your nominal resolution is coarse, you have excellent signal to noise, and say you can fit to shorter times, that would give you precision but not accuracy, right? So you could be subject to systematic errors in doing it. So having techniques which can give you finer time steps is always.
The laser basically, is on a pixel, for a very short time. And as long as, if we imagine imaging a pixel and then, walking the ultrasound stimulus at arbitrarily fine time scale, which is essentially what we're doing. And as long as we repeat for a long enough period of time, then we can get that fine information.
Do you want to take one more minute and tell people about your highest priority next steps?
Okay.
Don't have to.
So it's twofold, I would say. One is getting down to single cell resolution for purposes of again random access, being able to play the piano as Rafa said, arbitrarily activate cells, and we have a good understanding of cell types in retina, not complete, but I would say if any neural circuit is going to achieve the goal of recording the electrical activity of every cell and understanding it, it's probably going to be the retina first. So that would be one goal, a single cell random access resolution, and then the translational goal is we're going to be doing some in vivo experiments, but also as rapidly as possible getting to patient reports. Ultrasound is already used clinically to diagnose the back of the eye. So people already put ultrasound in the eye, likely with the different power levels, different protocols than we use, but be able to see if a patient can report if they're seeing flashes of something in response to ultrasound would be
Great.